Scientists have found the real Adam. He did not live in Eden. He had no garden and no forbidden fruit. He lived in a small village in what is now Western Cameroon. And here is what is more terrifying than any ancient book. Most of the male line branches that grew alongside him were cut off. Not by a flood, not by the wrath of God, by other men, his own descendants. This Adam was discovered by accident. An African-American man from South Carolina gave saliva to a commercial lab to find out where his great-grandfather came from. The technician looked at the result and did not know what he was looking at. The chromosome did not match any known branch of the human family tree. It was the deepest branch ever found in a living person. And as it turned out, that branch leads back to the man from whom all male ancestry began. But what came next was darker than any mythology. Who was he really? Why did his direct descendants almost disappear from the earth? Who pushed them out? And why are most men on the planet today descendants of a completely different man who lived much later? The one whose blood flows through most men in Western Europe. The one scientists call the second Adam. That man was named Albert Perry. He had long since died, but his descendants, the Perry men, were still living in South Carolina. In 2012, one of them sent a DNA sample to Family Tree DNA, a routine commercial test, the kind millions of people take every year out of curiosity. A woman named Bonnie Schrack saw the sample. She was not a scientist in the academic sense. She was a citizen researcher, a volunteer who had spent years working through genealogy. Schrack looked at the Y chromosome markers, the part of DNA passed only from father to son, unchanged for thousands of years, and realized she did not recognize any familiar combination. She wrote to Michael Hammer's team at the University of Arizona. Hammer was one of those scientists who had spent decades building the family tree of human male lines. When he received the data, verification took weeks, then months. When the result became clear, the paper was published in the American Journal of Human Genetics in March 2013. They named this branch A00, and they estimated its age. By their calculations, it was 338,000 years old. That meant the common male ancestor of all living humans had lived 100,000 years earlier than expected, earlier than the oldest skeleton of a known modern human, earlier than everything in the textbooks. But that was only half of the discovery. The next part took the scientists to Africa. If Albert Perry's line is that old, there had to be other carriers. The University of Arizona team took a database of 5,648 men across Africa, from Senegal to Mozambique. They ran the tests. At first, nothing. Then they found them. 11 men, all from the same place. It was Western Cameroon, a small Mbo population. Out of 174 samples, 11 had the same deepest lineage, 6.5%. Among all the other peoples of Africa, 0. Albert Perry had been brought to America through the slave trade sometime between the 15th and 19th centuries. His ancestors had been taken from the same West African coasts centuries earlier. The line survived the ocean crossing, slavery, generations, and surfaced again in South Carolina. So the real male ancestor of humanity, not a legend but a biological one, lived in the region where a few thousand people now speak the Umbeo language. They live in small villages in Western Cameroon. They know nothing about the fact that they carry the oldest trace of the planet's male line in their Y chromosome. And that is where the discovery ended. But the mystery had only just begun. Because A00 makes up less than 1% of Africans, an extremely rare line. If it is so early, so ancient, why are there almost no carriers left? That question was answered in Tartu. It is a small university city in Estonia. In 2015, a young researcher named Monica Carmen and her team decided to do something no one had done before. They collected 456 complete Y chromosomes from 110 populations around the world. High quality. Not fragments as before, but the full thing. When they built the map showing how these male lines split over time, they found two surprising things. The first one was no surprise. All male lines outside Africa split within a very narrow time window, about 50,000 years ago. That was the out-of-Africa migration. A small group of hunters left, settled Eurasia, then Australia, then the Americas. All as expected. But the second finding did not fit into any textbook. Among women, the line carried through mitochondrial DNA passed only from mother to child. There was nothing strange. Diversity rises smoothly over time, just as it should. Among men, a collapse. Deep, sharp, global. Between 5,000 and 7,000 years ago, male lines suddenly became much less diverse. There was no war to explain it. No epidemic. That would have affected women too. No famine. A part of the male branches was simply cut away. A Stanford press release that later described the work used a comparison that spread across the internet. It looked as if every man who fathered children had 17 women doing the same. 17 to 1. Whatever it was not a natural disaster. It was something that selected men. The explanation came from an unexpected place. Not from a scientist with 30 years of experience, from an undergraduate sociology student at Stanford. His name was Jianchen Zheng. He sat in a dorm room, read blogs about Neolithic Europe, argued with the authors, and at some point wrote to his high school friend. The friend's name was Alan Aw, spelled A-W. He was studying nearby in a mathematics and computational science program. They brought in Professor Marcus Feldman, a biologist. The paper was published in Nature Communications in 2018. Their explanation was simple. When people began to settle the land, something appeared that hunter-gatherers had not had before. Property. A field, a herd, a house, a granary. All of it had to be inherited. It passed through the male line, from father to son. That is how clans emerged, tied together not just by kinship, but by a shared Y ancestor. One great-grandfather, and we are all his descendants, all one family. Clans fought other clans. It was not individual men killing individual men. Clans erased other clans. When the defeated were slaughtered or driven out, it was not one life that disappeared. It was an entire male line. All the Y chromosomes descending from their common grandfather. All future descendants. A whole branch of the tree, cut off in one generation. Women often remained. The victors took them as wives. Their mitochondrial line continued. The male line did not. And so, over 2,000 years in the Old World, most male branches were simply erased. And that is where the story comes back to Adam. Only now, this is a different Adam. Among the surviving male lines, some behaved strangely. They did not just survive the catastrophe, they exploded. In a short time, one branch could produce tens of thousands of male descendants spread across vast distances. The most striking such branch is called R1b, and scientists know where it came from. In 2015, a large team led by Wolfgang Hack from the Max Planck Society's Institute for the Science of Human History in Germany and David Reich of Harvard Medical School published a paper in Nature that is now considered one of the major events in archaeogenetics. They analyzed the complete genomes of 69 ancient Europeans, covering 8,000 years, from early farmers to the Bronze Age. And they saw this. About 4,500 years ago, herders moved west from the steppes between the Black Sea and the Caspian Sea, from what is now southern Ukraine and southern Russia. Archaeologists call their culture Yamnaya. They had domesticated horses. They had wagons. They had that same clan order the Stanford researchers talked about, patrilineal, warlike, aggressive. When scientists looked at the Y chromosomes of Yamnaya men, all 7 samples belonged to the same haplogroup, R1b. More specifically, the branch called R1b-M269. That line moved north and west. Hack's study showed that 75% of the genetic material of the corded ware culture, The largest archaeological culture in Central and Northern Europe at the time came from the steppe. Not trade, not cultural exchange. A direct mass migration that replaced a significant share of the male lines living there before them. The Kurgan hypothesis, proposed by Maria Gimbutas in 1956, that the Indo-Europeans came from the steppe, finally received confirmation. 60 years later. From graves. But the most astonishing event did not happen in Central Europe. The most astonishing thing happened on an island. Neolithic Britain was a crowded place. Farmers had arrived there thousands of years earlier from the continent through what is now Turkey. They built the most famous megalithic monument on the planet, Stonehenge. They dragged stones weighing several tons for hundreds of miles. They built it over generations. In 2018, Inigo Olalde from David Reich's lab at Harvard published the results of an analysis of 400 ancient genomes in Nature. When the team looked at the Y chromosomes of Neolithic British men, the very people who built Stonehenge, not one of the 33 had R1b. Not one. 0 out of 33. A few hundred years later, people of the so-called Bell Beaker culture arrived on the island. They came from northern continental Europe, from the lower Rhine region. They brought with them the same steppe ancestry as the Yamnaya, and they brought R1b. When scientists looked at the Y chromosomes of British men from the Copper and Bronze Ages, 52 samples, more than 90% had R1b. In the same paper, Olalde wrote something that should make anyone who has ever walked past Stonehenge uneasy. Over a few centuries, about 90% of the entire gene pool of Britain's population was replaced. Stonehenge still stands today. The stones were raised by people who are almost gone. They may not have had horses or the rigid clan system that helped those coming from the continent win. They built an everlasting monument, and yet almost no male lines remained after them. A monument to the losers. That is why this story began with the real Adam in Cameroon. The first Adam lived 338,000 years ago in West Africa. He is the father of every Y chromosome in every living man on Earth, including every viewer of this video. His direct descendants, the Mbo people, now make up less than 1% of Africa's population. Almost all the branches that grew beside his line were cut off by other men, other branches of the same male tree. The second Adam lived only 4,500 years ago in the steppe. His name is not written in textbooks, and no one has seen his face. But his blood flows through most men in Western Europe. In 70% of Irishmen, 80% of Welshmen, 60% of Frenchmen. His descendants arrive, take the land, set their rules, and the lines that lose disappear quietly, without books or monuments. Between them lie 330,000 years. And in those 30,000 generations, there were far more male lines than there are today. Most of them did not survive. If Adam and Eve met, and according to the latest estimates, they missed each other by 100,000 years and thousands of miles, they would have seen people all around them. Many people. Their descendants from other men and other women. And almost all of those lines had already ended long before today. The Bible tells the story of one family. DNA tells the story of a catastrophe in which only the strongest clans survived. Not individual men, not individual heroes. Clans, alliances of blood brothers who methodically, generation after generation, erased everyone else from the future. That is the real truth about the first man. He was not alone. He was one of many. And the line that reached us, almost alone.